you're talking about a single-celled bacterium living in the soil, a cell in a plant leaf, or an incredibly complex neuron in the human brain like these beauties we see here, which are pyramidal neurons, every single cell on the planet is made up of just 4 different classes of biological molecules: the carbohydrates, lipids, proteins, and nucleic acids. They make up the organized structure of the cell, and their interactions are the basis for all cellular function. We're going to examine each of these classes of biomolecules in detail. In this video, we focus specifically on the carbohydrates. First, let's think about the term carbohydrate itself. These molecules are hydrates of carbon and will have molecular formulas that are some multiple of CH2O, N, where N is 3 or more. If I were to ask you what functions you associate with carbohydrates, you'd probably say energy, and you'd be right. Simple sugars or monosaccharides, mono meaning one, saccharide meaning sugar, are a major source of cellular energy. Monosaccharides like glucose are oxidized through controlled chemical reactions, releasing energy that the cell can then capture and use to synthesize ATP. But if energy is in excess, cells don't have to immediately use it up. They can store it for later use, and they do this by linking individual monosaccharides together to form larger molecules. Disaccharides with 2 monosaccharides covalently bonded, di meaning 2, and much larger molecules called polysaccharides, poly meaning many. For example, starch, with thousands of monosaccharides linked together that the cell can then access later when needed for oxidation and energy release. But carbohydrates are associated with other interesting cellular functions besides energy as well. Some organisms use other polysaccharide molecules to build structurally rigid molecular support, and 3 examples of this are the cellulose and chitin molecules that are the main molecular components of plant cell and fungal cell walls. Chitin is also used in crustaceans and insects in their exoskeletons, and while animal cells don't have cell walls, we do have a molecular network outside of our cells called the extracellular matrix, which provides supportive and other functions, and polysaccharides feature strongly in that matrix as well. Finally, many cellular proteins and lipids are modified by the addition of sugars in the form of oligosaccharides. Oligo means few, so these are polysaccharides with typically in the range of a few to dozens of monosaccharides linked together. These carbohydrate additions are important contributors to specificity of intermolecular interactions via their specific structural features and chemical properties. Now, to understand how carbohydrates can serve these diverse cellular functions, we need to investigate their structures, and we'll start with simple sugars or monosaccharides. Monosaccharides consist of carbon chains of varying lengths, and this is one basis on which they're classified. The most common sugars in the cell have 3, 5, or 6 carbon chains, and they're called trioses, pentoses, and hexoses. Now, in addition to the carbon backbone, what other functional groups can you identify in the sugars shown here? Hopefully you said hydroxyl and carbonyl groups, because they're present in all sugars. We know that these functional groups are polar, and so sugars are polar and readily associate with water molecules, making them hydrophilic. Sugars with carbonyl groups that are aldehydes at the end of the carbon chain are called aldoses, and sugars with carbonyl groups internal to the carbon chain are ketoses. Glyceraldehyde, ribose, and glucose are all aldoses, while dihydroxyacetone is ketose. For reasons that will soon become clear, we need to distinguish between the different carbons along the backbone, so they're numbered. And by convention, the carbonyl carbon, if it's an aldose, or the end carbon nearest to the carbonyl, if it's a ketose, is numbered 1. And the other carbons are numbered sequentially 2, 3, 4, etc. So if we're numbering the carbons in this glucose molecule, for example, this carbonyl carbon would be carbon number 1. This would be 2, 3, 4, 5, and 6. Now, glucose is by far the most common cellular energy substrate. We'll talk a lot about glucose in this class. Ribose and the related sugar deoxyribose are components of the nucleotide building blocks of DNA and RNA. Glyceraldehyde and dihydroxyacetone are both intermediates in sugar synthesis and oxidation pathways in cells. We'll see them later in the energetics unit as well. But let's focus on glucose, given its central importance for a lot of biology. Here's a question for you. We've talked about stereoisomers and chirality and enantiomers. Take a look at this glucose and see if you can determine how many chiral carbons there are in the molecule. Remember that in order to be chiral, a carbon must be bonded to 4 different atoms or groups of atoms. Pause the video now and evaluate each carbon for chirality. Let's look at each carbon individually, starting with carbon number 1. In this case, carbon has only 3 bonded partners, so by definition it can't be chiral. What about carbon number 2? Well, if you imagine yourself as that carbon and look down each of its 4 bonds, do you see 4 different atoms or groups of atoms at the end of those bonds? Now, remember to take into account the whole molecular group you see at the end of the bond, not just the neighboring atom. The answer is yes. Same with carbon 3, carbon 4, and carbon 5. But when we get to carbon number 6, the answer is no, because this carbon has 2 hydrogens covalently bonded to it. So with 4 chiral centers and 2 possible stereoisomers at each chiral center, how many different stereoisomers of this sugar are possible? 2 possibilities exist at each chiral center to the power of 4 because there are 4 chiral centers. So there would be a total of 16 stereoisomers. This isomer is called D-glucose, and it's the one we and nearly all biological organisms oxidize for energy. L-glucose would be a mirror image of this molecule, and the enzymes that initiate the metabolic breakdown of D-glucose for energy would not be able to bind to and act on it. From here on out, when I say glucose, I'm referring to D-glucose. Here we see the isomer of glucose referred to as galactose. It differs from glucose spatially only at the 4th carbon with respect to the arrangement of the hydroxyl group and hydrogen atoms. Galactose is used biologically. It's a component of milk sugar, lactose. So some of the 16 stereoisomers of this hexose are used and some aren't. Now what about this hexose on the right, fructose? It has the same molecular formula as the other two, C6H12O6. Is it a stereoisomer of D-glucose? Well, no. In this case, there's actually a difference in the covalent connectivity. Notice that the carbonyl group on fructose is internal to the carbon chain, so it's a ketose sugar. So fructose would be a structural isomer of glucose. These naturally occurring stereoisomers or structural isomers can be isomerized or rearranged by specific enzymes to produce glucose, which cells then oxidize for energy. So these are all energy-yielding compounds. These linear structural formulas make the covalent connectivity and orientation easy to see, but this isn't really what monosaccharides look like structurally in aqueous solution like we'd see in a cell. Instead, what we see is the linear chain folding back on itself and forming a closed ring. Now remember that each of these carbons with 4 bonds will have a tetrahedral molecular geometry. And also remember that these single bonds allow rotation. So what can happen is that these carbons along the backbone can each swivel around, and sometimes they can do that in such a way as to bring the carbonyl group in close approximation to a hydroxyl group towards the end of the chain. Hydroxyl groups and carbonyl groups will spontaneously react with one another to form a new covalent bond between the hydroxyl oxygen and the carbonyl carbon. Now again, this type of drawing is meant to provide a sense of perspective when looking at the structure. This thick wedge is meant to indicate that this part of the ring is coming out of the page toward you. Typically, if we're looking at an organic compound like this and there's no other atom indicated in the backbone, we just know that the atom at that position in the molecule is a carbon, because they form the backbone of all of our biomolecules. So to make this a bit clearer, let's number the carbons on the linear chain: 1, 2, 3, 4, 5, and 6. And likewise, let's number the carbons on the ring that is forming: 1, 2, 3, 4, 5, and 6. So you can see in this specific case, the interaction is between the hydroxyl group on the number 5 carbon and the carbonyl group involving the number 1 carbon. Other interactions could occur which would form different ring structures, but this is the most stable and most common way this happens. Okay, so we end up with a closed ring, but here's the thing. When this hydroxyl and carbonyl group interact, the new bond is formed between the carbonyl carbon and this hydroxyl. And we could simplistically envision this hydrogen being transferred to what was the carbonyl oxygen to now form a hydroxyl group on this number 1 carbon. So number 1 carbon goes from having 3 bonded partners, carbon, oxygen, and hydrogen, to having 4 bonded partners, carbon and the rest of the ring over here, oxygen and the rest of the ring over here, and then a hydrogen atom and a newly formed hydroxyl group here. So what we get is a new chiral center. And depending on the orientation of this carbonyl oxygen with respect to the rest of the ring when it closes, we're going to end up with two different stereoisomers. One in which this hydroxyl on the number 1 carbon is positioned below the plane of the ring, which is referred to as alpha glucose, and this version in which the hydroxyl is positioned above the plane of the ring, which is referred to as beta glucose. Now in aqueous solution, both of these ring forms are present and will freely interconvert back and forth, with the ring opening back up to the linear chain and then rapidly closing back up to form a ring again, which could be in either the alpha or beta orientation. The alpha isomer is a bit more stable, so it tends to predominate with a ratio of about 60% alpha and 40% beta, and a very, very, very small amount present in the linear form. This ball and stick representation presents another view of the difference in spatial orientation between alpha and beta glucose. Here are the carbon atoms in gray, 1, 2, 3, 4, 5, 6. Oxygens are red and hydrogens are white. So here's our ring oxygen and the others that are part of hydroxyl groups. Notice that the orientations of all of the molecular components are the same, except for this number 1 carbon, where the hydroxyl group and hydrogen atoms switch places three-dimensionally in space. This is a small structural difference, but it has very important consequences for larger polysaccharides that are assembled using these two versions of D-glucose. Cells with an abundance of energetic monosaccharide molecules can store those for later use. And to do this, they link single monosaccharides together to build large polymers or chains. Disaccharides are larger depending on the organism and cell type. For example, some plants will store energy primarily as the disaccharide sucrose, which is table sugar. Sucrose consists of a glucose molecule covalently linked to a fructose molecule. And the type of reaction that's used to do this is dehydration, in which the components of a water molecule are eliminated and a new covalent bond is formed between these interacting groups. Since we're talking about covalently linked sugars, this bond is called a glycosidic bond. The prefix glycol means sugar. This reaction would not occur on its own, but would require a catalyst. Biological catalysts are called enzymes, and they're nearly all proteins. So these two sugars would be recognized by an enzyme. The specific enzyme in this case is sucrose 6-phosphate synthase, in a highly stereospecific fashion, and the two hydroxyl groups from which the components of a water molecule are removed are on the number 1 carbon of glucose and the number 2 carbon of fructose. If we were to number fructose's carbons, this would be number 1, 2, 3, 4, 5, and 6. Don't worry, I won't ask you to identify any specific carbons in any sugar molecule except for alpha and beta glucose. The oxygen on the number 2 carbon of fructose remains and becomes bonded to the number 1 carbon of glucose. So if we were to describe the specific bond that was just formed, we'd say it's a 1,2 glycosidic bond. So what happens when the cell needs energy and wants to utilize its stores of glucose and fructose? Well, it would need to cleave this glycosidic bond, releasing the two monosaccharides, which could then be oxidized. This would be catalyzed by a different enzyme, sucrase in this case, and the reaction would be the reverse of the synthesis reaction. It would be a hydrolysis reaction, in which the components of a water molecule are added back in to cleave the bond, releasing the individual sugars. Other organisms store excess glucose energy as more complex carbohydrates or polysaccharides, commonly starch. Starch is a polymer built from linking together monomeric alpha glucose molecules in head-to-tail fashion to form a long polymer. Here are two alpha glucose molecules, and in the case of starch, the linkage that occurs is between the hydroxyl groups on the number 1 carbon of this alpha glucose molecule and the number 4 carbon of this alpha glucose molecule. So we call this a 1,4 glycosidic bond. A different cellular enzyme is required for this reaction, starch synthase, and what we end up with ultimately is a large molecule, a macromolecule, which is starch, a polymer of alpha glucose linked 1,4 in head-to-tail fashion, with each sugar oriented in the same direction, with number 6 carbons all facing up, for example. These polymers can contain tens of thousands of glucose molecules, so they're big. And once we have a chain of these alpha glucoses linked together, what will happen is that we'll start to have interactions between partially positive and partially negative charges in the polar constituents of these molecules, namely the hydroxyl groups and the ring oxygens all along the chain. And because of the specific structure of the alpha glucose molecules and the orientation of those interacting groups, the overall three-dimensional structure that we see forming for starch is a helical coil that looks like this. It looks kind of like a spring. Both plants and animals synthesize starch, i.e. alpha-1,4 glucose polymers. The animal version is typically referred to as glycogen, but it's built the same way as plant starch. In plants, there are two main types of starch, amylose and amylopectin. Amylose is a polymer that exists as a single chain with no sugar groups branching off of that main chain. Amylopectin does have some branching that'll look like this. And if you number these carbons, you'll see that these branch glycosidic bonds aren't 1,4 linkages, but rather 1,6. So different branching enzymes are required to catalyze the synthesis of these bonds. Glycogen, or animal starch, contains extensive 1,6 branching, whereas in amylopectin, branches may come off the main chain every 25 or 30 glucose units. On glycogen, branching occurs about every 8 to 12 glucose units. Nearly all animal cells contain some glycogen, but it's particularly abundant in muscle and liver. Up to 1% of a muscle cell's mass is glycogen, representing a reservoir of stored energy for that muscle cell to call on to meet energy demands as needed. Up to 8% of liver cells' mass is glycogen, and the liver can release glucose from glycogen into the bloodstream to feed the rest of the body when energy reserves are low. So liver glycogen is an energy reserve for the whole body. In plants and animals then, when glucose levels fall, these polysaccharides can be cleaved by cellular enzymes to release glucose monosaccharides that the cell can then oxidize for energy. Here's an image of a tightly packed glycogen granule like we'd see in muscle or liver cell. This is a protein at the center, but all of these branches are alpha-1,4 and alpha-1,6 polymers of glucose, so you can clearly see the extensive branching that's present in glycogen. Now, why do you suppose that there's so much branching in glycogen? Remember that functional requirements are a driving force for the evolution of structural features to meet those functional needs. So what's the selective pressure here? Well, if the function of this polysaccharide is to store energy and to release it when required, and the way that it's released is by an enzyme cleaving the ends of the chains to release glucose, we can see that having more branching means that we have more ends for those enzymes to work on. So in animals which have higher energy needs than plants, cells can release large amounts of glucose more readily when needed. Okay, so we know that starch is a polymer of alpha glucose, but what about that other isomer, beta glucose? Is it ever used for anything? Well, yes, but not by us and not for energy storage. Instead, we see beta glucose used in plant cells as the monomeric building block for cellulose, which is the main component of plant cell walls. Cell walls offer structural support and rigidity outside of cellular membranes, and they're found in a lot of organisms, but not animals. Remember that the difference between alpha and beta glucose is the orientation of the hydroxyl group on the number 1 carbon. When the ring closes, is it up or down? In beta, it's up, or on the same side of the ring as the number 6 carbon. These beta glucose monomers are also linked together by dehydration synthesis, involving the hydroxyl groups on carbons 1 and 4, but because we're talking about beta glucose, the way this happens is slightly different. This number 1 carbon's molecular orbital that's bonded to the hydroxyl is oriented up, whereas this number 4 carbon's molecular orbital bonded to the hydroxyl is oriented down. So how can these interact to form a new bond? Well, that's easy. We just invert one like we see over here. Now the spatial orientation is correct so that carbon number 1's molecular orbital is correctly oriented with respect to the oxygen on this carbon number 4 that it's going to become covalently bonded to. Now we can have that enzymatically catalyzed dehydration synthesis reaction linking the beta glucose monomers together to form the polymer, and we end up with the molecule cellulose, a beta glucose polymer linked 1,4. But notice that the orientation of these glucose molecules with respect to one another, they're still linked head to tail, but the orientation of each sugar is flipped 180 degrees with respect to the neighboring sugars. So the orientations of the polar functional groups along the polymer with respect to one another is not the same as what we saw with starch, and this structural difference is hugely important. The hydrogen bond forming groups are oriented differently and can't interact in the same specific fashion that they did in the starch polymer. And so the overall polymer doesn't coil up to form that helical shape. Instead, the cellulose polymer remains as a flat ribbon-like molecule. And since it's linear and flat and has lots of polar functional groups that can hydrogen bond with one another, what happens is that cellulose molecules will stack one on top of another, forming intermolecular hydrogen bonds with one another, and forming fibers consisting of numerous cellulose molecules, all hydrogen bonded to one another. This is going to lead to the functional property that we associate with cellulose, which is structural rigidity. This is a structural comparison of polymers of alpha-1,4 glucose and beta-1,4 glucose to make this difference in structure a bit more apparent. In these GIFs, we're just looking at two glucose monomers linked 1,4. On the left is alpha, on the right is beta. If we rotate these around, we can already start to see the difference in three-dimensional shape due just to the structural difference around that number 1 carbon. The alpha ends up being a bit more bent compared to the beta, which is more flat. So again, when we link alpha glucose monomers together, this bent configuration and the orientations of the interacting polar groups cause the overall molecule to bend around, forming a helix due to intramolecular hydrogen bonding. Here we're looking at that from the side, and here we're looking down the helical axis. But when beta glucose monomers are linked together, the polymer is flat and linear, and this leads to stacking and intermolecular hydrogen bonding between different cellulose molecules. And although individual hydrogen bonds are relatively weak, when you sum them over these giant molecules, they are collectively very strong, and so we end up with this aggregate that has a great deal of structural rigidity. Here's another view of the individual cellulose molecules and the specific hydrogen bonding interactions that we see between them. Note that the interactions are between partially positive hydrogen atoms in hydroxyl groups with partially negative oxygen atoms in other hydroxyl groups in another molecule. Plants arrange these cellulose fibers in an overlapping crosshatch network outside the cellular or plasma membrane to provide that needed structural support that allows a land plant to stand against the force of gravity. Other organisms follow the same basic principle, using polysaccharides for structural support, but built from slightly different monomeric building blocks. For example, using the sugar N-acetylglucosamine to build chitin, which is the main structural component of the cell walls we see in fungi, and of the exoskeleton that some animals have, including crustaceans and insects. Here's a polymer of N-acetylglucosamine. Here's the structural difference with glucose. Instead of this number 2 carbon having a hydroxyl group attached, it has an amino group with a nitrogen, and this is the acetyl. So this whole group here is the N-acetyl, just by way of explanation. These are 1,4 linkages as well, and we can see the structural parallels with cellulose in terms of this alternating up-down orientation of the N-acetyl group, which is also going to result in a flat ribbon-like molecule, just like what we see with cellulose. Therefore, this is going to lead to the same sort of extensive intermolecular hydrogen bonding that we see with cellulose, and the same sorts of functional properties in chitin. Like cellulose, chitin molecules form fibers, which are then wrapped in protein and arranged in highly specific patterns. Here we're looking at the arrangement in lobster exoskeleton, where the chitin network would be mineralized in similar fashion to what happens in our bones to give that strong structural support to the organism. But the same sort of crosshatch network of chitin fibers would be found in fungal cell walls as well. Animal cells don't have cell walls, but we do have less rigid, more gel-like molecular meshworks outside of cells in solid tissues that provide structural support and other functions to our cells, and this is called the extracellular matrix. In many tissue types, polysaccharide and protein aggregates called proteoglycans, proteo meaning protein, glycogen meaning sugar, in the extracellular matrix confer very specific properties to the tissue. As an example, let's look at cartilage. We know that cartilage has kind of a firm, rubbery consistency. We find it in our nose, in our ears, other places as well, but importantly also between the ends of bones, where the cartilage provides a cushion against impact forces that would otherwise see bone grinding on bone. If we were to zoom in microscopically to look at cartilage at the cellular and molecular level, what we'd see is cells called chondrocytes all enmeshed in an extracellular matrix that they've synthesized and secreted, made up of an extensive Proteoglycan network, which gives this meshwork of cross-hatched molecules like we see here. Now what this meshwork is made up of is protein, molecules like collagen, represented by these long fiber structures, fibronectin and various other link proteins that connect these proteins to these other structures made up of branching carbohydrate groups coming off of a central core protein, these little bottle brush structures. This is the Proteoglycan network. If we examine these bottle brush shaped molecules more closely, we'd see that covalently bonded sugar chains are comprised of glycosaminoglycan sugars, which are sugar molecules that have amino nitrogens and other polar or charged functional groups, most commonly sulfate. This is the structure of one of the most common glycosaminoglycans, chondroitin sulfate, but there are a number of others that would be present. So these are linked together in chains, and those chains are connected to a protein core, and that's what this bottle brush structure represents. Now, how is this molecular composition of this aggregate related to those properties of cartilage that we just talked about? Well, the sulfate group is negatively charged, and so it can attract water molecules very strongly. So these aggregates, being polar and actually charged, hold a lot of water, and water is not compressible. So this is what provides the shock-absorbing function of that rubbery cartilage between bone ends. Proteoglycan networks are maybe not as extensive in all tissues, but they're typically present to a greater or lesser degree. Finally, the last thing I want to talk about in terms of carbohydrates is glycosylation. Carbohydrates are frequently added on to proteins and lipids after their synthesis, and these modified molecules are called glycoproteins and glycolipids, and the process of adding those carbohydrate groups is called glycosylation. These carbohydrate groups are added enzymatically in highly specific structural patterns, and these have important influences on how molecules can then interact. On the right is an example that illustrates the kind of diversity we may see in glycosylation patterns. Each of these represents a monosaccharide, but notice that there are several different specific sugars represented here. Down here at the bottom in yellow is represented the amino acids in a protein that this branched carbohydrate network has been added onto. And in terms of sugars, we see fucose, glucnac, which stands for N-acetylglucosamine, mannose, more glucnac, galactose, and sialic acid. And notice also the diverse types of glycosidic bonds that may be present: alpha-1,6, beta-1,4, alpha-1,6, alpha-1,3, and so on. So lots of structural diversity present in terms of the types of sugars that are present and how they are covalently bonded to one another, generating a lot of diversity in terms of overall structure and the orientation of the polar, hydroxyl, amino, and other functional groups that are going to be present on these sugars. We frequently see this type of glycosylation on proteins and lipids in cellular membrane, and I'll just cover a couple of examples in terms of their important functional contributions. The first is in cell-cell recognition with regard to immune defense. Obviously, if a multicellular organism is going to defend itself against foreign pathogens, it has to have some capacity for non-self recognition to prevent autoimmune attacks. The interactions between immune cells and the body's own cells frequently are mediated by glycosylated proteins at the cell surface, and the specific branched carbohydrate groups are what provide some of the specificity of that interaction. For example, the ABO blood groups actually describe just these sorts of glycosylated proteins. The A, B, and O designations describe different branched sugar groups that would be present on the surface of red blood cells and other cells in the body. So when someone is transfused with the wrong blood type, their immune system will not recognize those foreign sugar groups and will mount an attack against those foreign blood cells, causing a dangerous transfusion reaction that can be lethal. Frequently, cell surface receptors are also glycosylated, and this will influence the types of chemical signals that receptors can interact with and respond to. The field of glycobiology, which is focused on glycosylation and its functional significance, is relatively new. And this field has a lot of clinical relevance for a lot of reasons, but one big reason is that infectious pathogens will frequently mimic the natural molecular partners of these cell surface molecules, and they'll bind to these carbohydrates. This type of interaction is often associated with the mechanism of infection, in other words, how the pathogen gains entry into the body, or otherwise exerts its harmful effects. In this video, we reviewed some major biological functions of carbohydrates. We've investigated the structures of monosaccharides and polymers of monosaccharides, and we've related the structural properties of some important polysaccharides to their functional significance. In the next video, we'll take the same approach, focused on the class of biomolecules called lipids.